A6. And Moses is saying that video is starting his own.
Hi guys. Professor. Good morning. It's Sam already here. Okay. Good morning, Dr. Sam.
Hello, sir. How are you, sir?
I'm fine. How was your night?
That's good. Yes.
Yeah, welcome. I think we have to start pushing already. We are 4 minutes late.
Yeah.
Yeah. So maybe a little feedback from our people, then we could move on. So,
Okay.
yeah, so I open the meeting in the name of the Father and of the Son and the Holy Spirit. Amen. So, I don't know if there's anybody that have a news or a feedback before we could go on.
I'll be back. Just let me get it. Something. I'll be back now.
Okay. Morning, Professor. You are muted. Your stuff is here, yeah.
Good morning, Dr. Moses. I'm doing adult class.
Yeah, it's wonderful. It's wonderful. I'm not dog in a trouser. Don't dog in all
the too much. When I know how. At my own time, I will go through again and they teach myself.
Amen. As well. Are you in the on-site now, or?
I'm in my office, private office.
Okay. The internet there is okay.
Yes, at least for now, it's working.
Is it personal internet or university internet?
University internet at the admin block B.
Okay, B, yeah. Usually they have good internet there. Yeah, but sometimes maybe towards afternoon. It might shake a little. Yeah. Okay, so we take a little feedback from yesterday. I think the assignment was to fetch 2 proteins, play around these proteins, understand the amino acid positions. What again? Yeah, look out for chains, present chains, whether chain A or B. Then I think finally was to superimpose these proteins together. I think that was just it. So we might need feedback from, maybe I start with Joy Geneka, because I know she should have something. Joy, I'm asking you to unmute now.
Good morning.
Morning, how are you?
I'm very well, thank God.
Oh, praise God. So how far? How did it go?
It was so fine. I did it, but I'm using 2 systems. So I don't know if I can't really share my screen now, but I'm using 2 systems. Yeah, I don't know if you understand what I'm saying.
Yeah.
Yeah, but I did everything and it was so good.
I understand. You connected one and using one to practice.
Yes.
Okay. Okay. But so far you are quite updated for today.
Yes.
Okay. Yeah,
yes.
So I moved to Lagos, Bankole. I ask you to unmute.
Good morning, sir.
How are you, Bankole?
Great. Let us
know. So how did it go with you, or how it went with you?
Yes, I was able to go through the assignments, practice all that we learned yesterday. At least.
Were you able to superimpose?
I did.
Yeah. So what we shall learn today is also a tool that will help us to know whether these things are properly aligned. Just as when you do BLAST, you could see 100% identity, 90. So after superimposition, it's also good to interpret if we had 100% superimposition. So when Doc will start today, I think that is part of the things we shall get to know. Yeah.
Oh, that's great.
Yeah.
That's great.
Okay, thank you. So I move to Akure. Atanda Halimat, I ask you to unmute.
Hello, good morning.
Morning, how are you?
I'm good, thank you. How are you?
Yeah.
Yep.
Are you from Igbo land? Sorry.
Yeah, I'm from Enugu State.
Enugu, okay. Harlem, okay. But you are born in Ondo.
Osun State.
Osun State, okay. And your father is from there?
Yes.
Oh, I see. Because the name just messed up. Okay. So give us good news.
Yes, good news. I went through the video because I wasn't here yesterday, and I did everything except for the superimposition. Well, I did it, but I don't exactly know I don't know what I did exactly, but I know I clicked some things and I did the superimposition. I got some sequences as results.
Sequences? Do we supposed to get sequences? Okay, so maybe to superimpose is another way of saying to when you are working with only sequences, yeah, you could align. So
okay,
sequences together to see how whether they are fitted or they have some discrepancies along the chain. So to superimpose also using protein, especially the quaternary. Now, it's like saying to align, just to put the 2 things and see if they are the same. And today we shall learn how to interpret if they are 100% fitted or 80% variation. So this is a means of understanding what we call selective site. For example, if you want to target a Plasmodium falciparum in human. So this is a way of sorting out the site you could target in Plasmodium that you will not affect in human. Okay, so when you put the 2 proteins together, you will see the regions that are not that is in Plasmodium but is not in human. So that particular site now, it could also be an important site to target If not, your drug could kill the parasite and kill the human at the same time. So these are the principles behind some of these things. Okay, but a good homework. Yeah, what is important is to do homework. So thank you. As I move to Chikodele Anugu, I hope she did something. So Chikodele, I've asked you to unmute. I'm coming, James. I'll come back to you later.
Hi, Moses. Good
Hi.
morning.
Morning.
Yeah, I did the assignment.
Mm-hmm.
I looked at the protein.
Mm-hmm.
I also spiked.
Okay.
So
it
was beautiful.
was beautiful.
Yeah.
Okay. Thank you. Happy and encouraged to hear that from you. Okay. Edith Bakare.
Good morning,
Morning.
everybody.
How are you?
I'm fine.
Yeah.
I did the assignment, checked for protein bonds and others.
Okay. And everything was perfect.
Yes, it was.
Okay. So I'm happy we shall end so well today. Okay. Thank you. And I've not gotten any feedback from Ihe Wumere Chidiogo Marigold. So I ask you to mute and give us feedback. When did I keep? Marigold. Ihe Wumere. You know, she just eventually left the room. I'm mentioning her name. Okay. Onochie Chiamaka, pharmacist. I ask you to mute. Yeah.
Good morning,
yeah.
everybody.
Thank you.
Well, I tried. Can you hear me? Okay.
Yeah, it's important to try.
Yes.
Go ahead.
Yes. I tried assignments, but I didn't really get the thing very well. I don't think I did it very well. So I
You
think
repeated,
someone needs to retouch the superimposition thing for me.
Okay. Yeah, okay. It's also very straightforward, but maybe as Doc might review and recap there, you can catch it again. Okay. Thank you.
Okay.
Grace, you have not talked to us. Elemo Grace, I ask you to mute.
Yeah, good morning.
How are you?
Can you hear me?
Yeah, perfect.
I'm good.
Yeah.
I wasn't able to do the assignment because I wasn't following up earlier on. I have issues with my system. So it was just yesterday. I didn't participate on the first day. And I joined yesterday or later. So I wasn't able to do the assignment.
Did you see the link I sent on the website for your?
Yeah.
Yeah. Did you try going through them?
Yeah. But I didn't finish all. You know, it's very lengthy. So I downloaded the videos for the first and the second day. So I think I know when I'm done with them, I'll be able to do something.
Okay, it's okay. It's okay. So the WhatsApp might also be where we are. We can keep it for a week or two. Then okay. After which we can shut it down. So in the course of that, you can still raise some questions, ask some questions there. The facilitators will still be there to help you.
Okay.
Well done.
Thank you.
Yeah. I know Lillian is not yet connected with her audio. So no alone, Vivian. I ask you to mute. Okay. No, hello, Vivian. She's not here. Well, who is Chifo? Chifo Diaka. I asked you to mute. We have not heard from you at all in the course of the meeting.
Yeah, hello.
Yeah.
Good morning. Yeah. Good
What's
morning.
happening from your side?
I was having some network connection problem yesterday, but I'm following closely.
Okay.
I'm following the principles of the whole thing, and it's very interesting.
Did you do the assignment at all? Did you play around the protein?
I played around, but I struggled with it, and I didn't get to do it. But I'm following.
Okay, but you could download the video or the recording of what has been done Wednesday and Thursday.
Yes.
Okay.
Yes.
Okay. Okay. Wish you well. Yeah. You know, Sir Benjamin, you have not talked to us since yesterday, so give us feedback. You know, Sir Benjamin, give us feedback. Yeah.
Hello?
Yeah, go ahead. Yeah, we could hear you. Go ahead. You know, sir.
I'm not hearing you, sir.
But I can't hear you. I can hear you. It's okay.
Okay.
Yeah, go ahead.
The network is bad here.
It's not bad. I can hear you.
But I'm not hearing you. Just.
Why?
Your voice is fading. I'm not hearing you clearly.
But you're responding to me. Just give us update of your own assignment.
Sir?
Give us update of your own assignment.
Okay. Can I share my screen?
Yeah, perfect. I enable you now. So yeah, go ahead.
Okay, let me get to.
I have enabled you to share your screen. Yeah.
I was.
We are waiting for you to share.
Okay, sir. I'm trying.
Weißt du, wo die transparenten Kleber sind fürs Labor? Also sie sollten da sein.
Ja, also ich habe alle Drama-Marken.
Okay, wie weiß?
Ja, aber ich weiß nicht, wer sich da bedient.
Okay. Ah, ja.
No, sir, we cannot spend the entire day waiting on you
The system
to.
was full. It's not responding to me. My system was full.
Okay.
Let me open it again, sir. Let me.
Yeah, maybe you can do that. We want to now move on to start for the day. Yeah. Yeah, our resource person, today is his birthday, so he has some other plan. Okay. So maybe Sam, I could enable you to share your screen and start all over. You know, sir, we may not wait for that, for all of that.
I thought you were going to come back to me.
Okay, James, sorry, go ahead.
Okay, share my screen now. I did the assignment. Share my camera view. I don't know if you can see it now.
Yeah, I'm seeing, but it's not too clear.
Why is it not showing?
It's showing CAV.
It's showing what? Can you imagine? Should be network issues.
Yeah, I think it's
Okay.
network, so it's blurred. Yeah, but do you
I
have
did
any?
it.
You had any
I
challenge?
did it. My challenge, I was in I was on site when the explanation was going on. So identifying the gene, I couldn't identify the genes, but I did the superimposition. I understand that the enzymes from further research, there are cytochrome P40 enzymes and proteins. And I was able to superimpose, and I deleted some chains and I left 2 chains that superimposed. And I also did the presets. I colored and identified the ligand. I noticed that identifying the ligand, that is from structure, when you go to select and go to structure, when you go for the ligands, I don't know which of the superimposed protein is actually highlighted there. That's what I didn't really understand. So if it was both ligands that were highlighted, I actually highlighted and colored them a different color, but this stuff will just not show. I don't know.
Yeah, so it's okay. I think it's a good push from your side. Then please, you may need to go through the recordings yesterday. The identify chain is a click away. So go through the recording, then you can easily identify chain. But maybe if in the course of today, that also happens, that is fine. Yeah, just as I announced, Dr. Sam need to start immediately. Of course, some of us already wishing him happy birthday in the group. It's not bad anyway. So, but he will share his screen and move on from there. So thank you, and the rest that could not give feedback, we'll see how it goes in the course of the training. So you're welcome, Sam.
Okay, sir. Thank you so much. God bless you.
But
appreciate
Sam,
your greetings.
we shall interrupt in the course, because once we have the maximum number, we shall quickly take our group photograph. Yeah, quickly. Yeah, but not now. Monitor the number. We are 20 now. Maybe if it gets up to 22, 23, we can do that together for the purpose of record. Thank you.
All right. Okay. So I appreciate the feedback. I just want to go back through that superimposition, and I'm happy that so many people are able to get the visualization right. Okay. I assume you can see my screen. So I have my camera open, and I want to repeat that process of superimposition. So yesterday we had 2 proteins, 4D7D and 4A1R. So I'm going to open the 2 proteins now. And the other one I will just fetch 4D7D, 4D7D. Okay. So this is where we started from. Once again, I just want to emphasize this exercise is just to sample an hypothesis that you have 2 proteins and you have mutation in one of the proteins. And you want to find out if that mutation is translated to the 3-dimensional structure of that protein. That is the whole idea. So when you want to do yours, if you're interested in that kind of study, you can. Those are the kind of hypotheses you can check. So, but these are similar protein, but in this case, they were submitted by 2 different research groups. So to do the superimposition, and I went to Matchmaker. So again, I just repeat that tool, structure comparison matchmaker. And you need, on your reference side, you have the models that are on the display. So you choose one of them as a reference, and on the other side, you choose the other as the structure to match with the reference. Okay. Yesterday, I'm not sure I showed you, if you go to the last bit, it says after superposition, compute structure-based multiple sequence alignment. Okay, now that is the parameter that will help us know, give us a metric, as it's a number that will kind of give us the level of similarity between these two 3D structures, these two proteins. Okay, so just make sure when you repeat that, you have this checked. Okay, and that's all you have to do. And you just click OK or apply, as the case may be. I'm just waiting for it to.
Yeah, it's
It's done. So why is it giving me this warning? I don't want to do this. Oh my. Okay, I see what this person was talking about. Oh, see, this is a sequence showing the alignment, but this is between two chains. For those that are interested in alignment, you can see the similarity and where the gaps are in the two chains, but that's not where I'm going. So I'll just repeat that again. Okay. Just watch at the bottom of the screen, you will see a metric that will come out called RMSD, which is your root mean deviation. Okay, all the 455 pairs of atoms in these structures. It compares them and gives a metric of 1.314. And the unit is usually Armstrong. So it's basically saying, if I put this together, how far apart are the atoms in one structure to the other? So the greater the, what's the word I'm looking for? The RMSD value, the farther apart the atoms are, and that means the structure is not aligning. So a very high RMSD means the structure is not aligning. And if you have it in the neighborhood of 0.5 and less, that means it's almost close to perfect alignment of those structure. So that's the way you can, there are other tools that you can use. PyMol does alignment very well, and some other tools. But PyMol and Chimera are the ones I have used often to align 3D structures and to answer the kind of questions that I post now. Okay, I think that should answer the question and clarify the feedback. Any question or clarification? So I want us to get into this stuff. If there are no questions. Where's my chat box?
So we can deduce the rate of similarity through the sequence. Could we also do that?
Sorry, I didn't hear that.
Could we deduce the the percentage similarity via the sequences. I mean,
yeah, that's another. You can do that.
Okay.
You can align sequences. You look at the sequence.
Yeah, of the both chains.
I tried to do that yesterday, but I don't want to confuse the issue. Yeah.
Okay.
I didn't want to confuse issues yesterday, but yeah, you can do that. All right. I think I need to shut down my camera.
You need to restart even the system.
Yeah.
Yeah.
Camera is freezing. Okay, I'll restart it. Let me just start with the slide first. Let me just shut it down.
Okay, so guys, a moment so that the system could be started, then we go on. Yeah.
Okay, so if, I don't know how many people are muted, if you can just mute yourself if you're not talking.
Virtually everybody is muted.
Okay, cool. All right, so this, I just want to set the tone for this discussion by giving a brief overview of what molecular docking is all about.
There's a question from Chikodley.
Okay,
I have
so
a question.
I need to change. I think I need to.
Are you restarting the
change my
system
connection.
eventually?
Yeah, I'm even changing my connection. Thank you. Yeah, what's the question? On the
So
chat.
she said, I have a question. Yeah, it's on the chat. Maybe you can read.
Okay, let me check. Okay, that is right. Yeah, that is right. So if you have multiple chains, because the connection between the chains are not actually covalent bonds, they are hydrogen bonds. So it's difficult for the algorithm to actually take the whole system and superimpose. Okay, somebody said it's as though the alignment is not on the entire structure.
It's still on the same question.
It's the same question, yeah. So yeah, so it's sometimes difficult for some programs to do that because you can't hold those complexes together as a unit, as it were, to do that. But I'm not sure. I think there's a particular. I'm not sure about PyMol. I will check. You can check PyMol. You might be able to do that. Yeah, but most times when you have those multiple chains, the chains, especially if they are dimers or tetramers, they are the same unit. So what you observe in one can be can be extended to the other chain, as it were. Okay. Like I said, I didn't want to get into alignment, but I want to refer you to the manual on Chimera. Very simple to engage with, you know, and you'll be able to see how that is done. You can call up the sequence of each one, and just there's a tab that will say align sequence. So if you go to tools sequence and you call out the sequence, it will ask you the sequence of two models. You say yes, and you just say align sequence under that tab. It should do that for you. Okay, what is the significance of two proteins that do not align? Okay, I don't know your background, but I can speak from the perspective of medicine, in the sense that, for example, we have an enzyme in the liver that is responsible for metabolizing xenobiotics, and anything that any chemical that comes into the body. But we observe that, and drugs, of course. But what we observe is that some people have higher ability to metabolize some drugs than some other people. And if you take a close look, you'll find out that there's a mutation in that particular enzyme that enables people to be what we call a fast metabolizer, and some people slow or poor metabolizers. So it translates to the activity of the enzyme. There's a paper that looks at CYP3A4 or CYP2D6 or CYP3A4 functional activity in the metabolism of lignocaine, and they were able to show that those different mutants actually have significantly different activity when metabolizing that particular drug. Okay, so that's the clinical significance, because you might not need to change the dose if we're able to find out if a particular person has a particular mutation. You might need to change the dose, and that's the basis of pharmacogenomics and personalized medicine and the rest of them. Why do we do docking? Okay, we're going to get into docking now. So I
want to do the docking now.
Okay. All right. So my objective here is to get you acquainted with the process and best practices in molecular docking. We're going to use Autodoc Vina that is present within Chimera. Autodoc can be used on its own in so many other platforms. But I'm just going to use this simple one. So most biological properties of a protein depends on its interaction with other molecules. And we know that biological processes in cells actually depends on the interaction of enzymes with their substrate to catalyze a particular process, or protein interaction with a protein to set up a signaling process, or protein interacting with a nucleotide. When you think about the DNA polymerase in the process of transcription and the rest of them. So it's all full of interaction at the molecular level. Okay. Substances that bind to protein are called ligands. Even if it's a big protein binding to another protein, they are referred to as ligands. Basically, it means that something that's nature has designed specifically to interact with a particular protein. Okay. Now that interaction usually is a transient process. What I mean by it is just something that happens temporarily in order to execute a particular process within the biochemical pathway. So the interaction is maintained by weak covalent, non-covalent bonds, because covalent bonds are more or less permanent. You will take a lot of energy to break them and form new bonds. But these are weak non-covalent bonds that are involved in this kind of interactions. But I won't say that some of those bonds are hydrogen bonds, some are electrostatic or ionic bonds. Van der Waal, you can just Google these terms if you are not familiar with them, so you can get an idea of what they mean. And hydrophobic interactions, okay? So you have a binding site of a protein and you have this ligand that nature has designed specifically to bind or interact with this protein. And it forms those non-covalent bonds. Then once it's done with this process, the protein can release that ligand, as it were. Okay, but the effective interaction requires that many of these weak bonds should be formed. If the few bonds are formed, that interaction might not be strong enough to execute the desired process. Or if you have something competing with that substrate. So in pharmacology, especially pharmacodynamics, we speak about competitive and non-competitive interactions. Okay, so if something else is competing with it, it might be displaced from that by binding site, by something that can actually form more interaction with that particular binding site. Okay, so that's the basis. We know macromolecules interact. Now, molecular docking is a way to simulate that process. So essentially, I would define it as an in silico, or what we call a computer-based approach that aims to predict the binding mode and the binding affinity of a complex formed by two or more constituent molecules with known structures. Okay, keywords that I want to emphasize here. The binding mode refers to the position or the orientation of each of those molecules to each other. The binding affinity refers to how strong that interaction is between those two interacting molecules. And of course, we can only simulate if we know what each of them look like. So this molecular docking is part of what we call structure-based drug design. And because we know the structure, then we can apply this kind of techniques. If we have some compounds, some drugs we know works because we tested them phenotypically in rats or in cell cultures, but we don't know what they are interacting with. You cannot apply this kind of study in that case. Okay, it's difficult. We need to know, and one of the things molecular docking also does is it helps you to elucidate the mechanism of action of a particular drug. So one of my students was able to do that using what we call high throughput cross docking. We know this compound works, but we don't know how it works. We know it interacts with Staphylococcus, but we don't know how it kills the bug. So we selected all the proteins in Staphylococcus and tested the compound against all of them to find out which one does it interact with best amongst all these proteins. Okay, so that's one question you can answer. What we call it's called target fishing. So you can use it to kind of get an idea of what's the target of your ligand or of your drug, and it gives you an idea of the possible mechanism of action for that particular compound. So in terms of process, it involves prediction of a ligand conformation. So what I mean is that I just define what a ligand is, something that binds to a protein. And the question is, in which orientation does it bind? So the computer needs to find the best position, the most likely, not the best, the most likely position that the ligand will take when it enters the active site of the protein. That's the job of the algorithm, okay? Based on some parameters, which includes the global energy of that ligand. I'll talk about that now. Just hold on to that term. And secondly, it needs to predict the position. First is the conformation of the ligand, then the position of that ligand within the protein when they interact. Then it needs to now assess how strong is that interaction. So first, the conformation of the ligand itself, just the ligand standing alone. How are you? Because we are talking about molecules, like if you think about methane, CH4, with your C in the middle and your H coming out in that 4 bonds that are attached to an hydrogen bond, an hydrogen element. So the question is, what is the orientation of that compound in space? So that's what you refer to as conformation, and there's a particular conformation that the compound is comfortable, that you can say that this is the comfortable conformation for a particular compound. It can assume any conformation. I can stand up, I can sit down, I can kneel down, I can lie down, but these are different conformations that I can take at a particular time, But the question is, the computer needs to now predict which conformation which conformation will allow the system to use the minimum amount of energy. That's what it's trying to look for. That this conformation that is such that the system does not need so much energy to be in that conformation. Okay, so that's what the job of the algorithm. That's what it needs to do. And just to say that in many cases the binding site is known. before we start docking, we know where we want to go, and then we try to find a thing for that position. But having said that, there's something called blind docking, where if you're not sure the binding site, not that we're not sure, but we know that in reality, the compound will go to as many sites as it sees on that protein. That's in reality, that's what will happen, even though you are interested in how it interacts with the binding site or the active site. So what I put here is some questions that you can use docking to answer. How does your molecule bind? What is the mode of action of my molecule? I just spoke about how you can use it to determine the possible target for your compound if you find that this compound is having an effect on a cell, on a particular system, or on an organism. And sometimes, especially when we talk about drug discovery, you have a series of compounds that you want to test. Just before you start synthesizing 100 compounds and wasting a lot of chemicals, it's important for you to say, okay, let's go to the computer. Let's predict which compound is most likely to will interact with this particular receptor. Then we can select the ones that are most likely. Then perhaps we can spend money on that. So, and that's the birth of what we call a rational drug design. So rather than just screening thousands of compounds, and we know we can't do that in Africa, and it's very, very difficult. We don't even have the robotic system for high throughput. To synthesize a whole lot of compounds is very difficult. So one of the things we do is to screen virtually using molecular docking. And that gives you an idea of compounds that are what we call top 8 compounds that are most likely to bind. Then you can pick those compounds and synthesize them, then test them in vitro test or enzyme binding assays.
So sorry, before you go ahead, so there could be, from the previous slide, if I understand, then the understanding of the binding site before docking would involve a detailed study of the protein and possibly the druggable sites in the protein.
Yes.
Yeah. So in that, let me just, in that case, what does it involve to get a clear site where you want to dock?
So if you have a knowledge of the protein, like I said, especially when you look at and this is where UniProt is important, you must do a research on your protein before you bring it. UniProt tells you the domains of it can take you to helps you to study the domains of that particular protein. And once you have an understanding of where the domains are, and the domains might be give you an indication of where the binding site is. There are other softwares, SiteFinder, and the rest of them that can help you find or kind of predict the particular possible site for that particular protein. So that's another way. And but most of the time, if you look in literature, they will tell you the sequence or that constitutes that particular binding site, and that you can look for that even in your visualization. If you bring out the sequence from your visualization and select that set of enzyme that has been maybe conserved over the years, usually binding sites the residues or the amino acid in the binding site are well conserved, and that gives an indication that this organism or this system needs these residues to be the same over time. So it's using them for something, So those are the ways you can get an idea. Okay, is that okay, sir?
Yeah, thank you.
Yes, sir. So I've been speaking about algorithm. Algorithm basically means a series of code that executes the molecular docking. So it's the thing that is happening in your computer right now, that you are just watching the screen, but there's something running behind. Okay. So some people sit down to write the program that runs behind that. And so what they have done over the years, that we find out that the algorithms have improved over the years. In the past, we are able to simulate what we call a rigid receptor and a rigid ligand. So what it means is that you keep the receptor. It's like a drawing of the receptor, a drawing or a cartoon of the ligand, and you bring them together and try to see, okay, how possible, what is the possible position that they can take. Nothing is moving, very rigid. But we know in nature that is not the case, because this macromolecule and small molecule.
Taking a lecture.
Yeah.
Sorry. Yeah, I just wanted to ask if you're planning to be here on Sunday around 11.
This week?
Yes.
No. Yeah, I'm only here tomorrow.
Okay.
If you wanted me to help you.
Yeah, I need someone to just take something out
Yeah.
of an incubator and put it into a freezer.
Yeah, I
I'm
plan
looking.
my ELISA today, tomorrow, and I rest Sunday. Come back Monday.
Yeah.
Or maybe David can drive and do this.
No, I like theoretically I could do it as well.
Yeah, another
Honestly,
person that comes on Sunday.
Yeah, or maybe Srinivas also.
No, most I
Srinivas.
come for visits.
No, it's fine. You don't have to come extra. Yeah, it depends.
David wants
on
to do
the person
this.
who's already
Du
here.
kriegst du schon die ganze Zeit. Es ist niemand da.
Dann ist es vielleicht schon viel. Das noch nicht alle.
Okay, das mach es halt.
I'm not sure if I can continue.
I'm sorry, I've muted already.
Okay, thank you. All right. So I was talking about the algorithms that are designed to execute this simulation. So the first sets were designed in such a way that the receptor is static or rigid, and the ligand is also rigid. And they are very fast processes because, yeah. But we know that in reality, these systems are not rigid, they move. They are very flexible, as it were. So it improved, and it was able to, we are able to make the ligand flexible because they are small molecules. So we can achieve that, but it's going to cost us more time in terms of the time it will take for the process to be executed, but the receptor remains rigid. But in recent times, we have achieved some sort of flexibility with the receptor, specifically at the binding site. You can simulate it such that the residues or amino acid at the binding site are moving, and the ligand is also moving. So it's a dance between the two of them before the marriage takes place or the interaction takes place, which is more realistic than what we had before, you know. And we are now getting closer to what actually happens biologically in the system, and that gives us more reliable prediction of the interaction, and you can actually depend on the results or have confidence in the result that you get from that. This is a timeline that shows what has happened from rigid docking to semi-flexible to flexible docking. But now these days, we've gone beyond this now using artificial intelligence to actually predict interactions. Okay. So in terms of interaction, the thing we want to, the stages of molecular docking are basically post-generation and post-selection. Post-generation involves the process I spoke about, in which case the ligand is dancing towards the target, eventually finds its position, and the algorithm needs to find the best position, you know, that will require minimal energy for this ligand to interact with this protein. Okay, so this is a plot of the energy required and the conformation of the ligand and the bound protein. So what it does is it brings a ligand into close proximity with the target or with the protein and looks at the energy of the system. If there's a lot of entropy, if you remember, I don't really want to get into this, but there's a relation between your entropy, your enthalpy, and what we call the free energy. So you can just review that, your basic. You don't have to know it, or you can take my word for it, whichever one is you want. But it calculates the free energy of the system. If there's too much entropy, it feels that interaction is not realistic. It tries another position, another pose, and checks the energy, tries another pose, until it gets a pose in which case the system has come to a minimal energy level. Then it tells you that, oh, fine, at this position or at this pose during the interaction, it seems it requires minimum energy. Because it requires minimum energy from the law of thermodynamics, it means that the interaction can take place almost instantaneously, if you remember your thermodynamics. Okay, that's what it tries to predict. Is there a question? Let me just see quickly.
So it's asking how to determine the binding site before docking ligand into it. We talked about it, yeah.
What about it? I'm going to just put something up. There's some other online tools that you can use. That you can use to find it, yeah. But if you look at PDB, if you have a protein that has a ligand in it, that particular ligand is in the binding site. But I'll show you when we do the hands-on exercise now. Okay. Okay. Now the purpose of this slide is not to, okay, so ignore everything that you see here. Just focus on, we need to find a way to score. We use the word score. Basically attach a particular number, a numeric evaluation to the interaction that we see. So the computer tells you that this interaction in this position is the minimum energy, but it's also possible that your ligand takes this other position. It's possible that your ligand also takes this other position. So it gives you several positions, or what we call several poses of the interaction between the two systems, the two interacting moieties, the protein and the ligand. And as it does that over time, it calculates kind of a way to estimate the affinity between the two of them. So it considers the electrostatic interaction, the Van der Waals interaction, what we call the solvation interaction. You can read more about that if you want to, but if you don't, it's not really important for you to really get the details, but it will be important for you to know the forces that are involved in the score that you get from a particular software, for example, so that you know what are the constituents of the final numeric value that you get when it tries to give a score to that interaction. So it is important to note that. Okay, so if I just take a look at this, okay, let me just take this out. I'm trying to look at some of the processes that are involved. Just a minute. I think I would leave my screen like this. I'm struggling to move. That are involved in your receptor preparation. Okay. Depending on the docking program that you are using, you need to select your protein. And we'll look at that today. You can select from PDB. You need to find the site. We just spoke about site selection. Then you need to add charges to this protein. Basically, what you're doing is called, we call receptor preparation. Because we know these things carry charges, but what you have in PDB, they are not, I'm trying to look for the word now, because they are resolved, there are some issues with those structures, okay? And those issues need to be resolved before you can have an accurate simulation of your docking. Some of them will carry charges that are not right. I mean, for example, you might find a carbon with 5 bonds, and that you begin to think that is not possible. Okay, so that needs to be sorted out. So within the docking software, there are processes to prepare your protein that takes care of these issues and correct, add correct charges. Sometimes add all the hydrogens atom so that everything is accounted for. It removes water or cofactors. And we saw a cofactor zinc the last time when we explored a particular protein. And then it does a refinement. The refinement is basically what we call energy minimization. So energy minimization means that I need to get this protein comfortable before I start simulating the interaction with the ligand. That's basically what minimization. Resolved in a position that is non-natural. If you see me, is it possible? Trying to walk with my hands and my legs are up, that's not a very natural position. If you see a picture like that, you will begin to imagine there's something holding my leg up, even if it's not showing. Okay, so you need to minimize my structure. So you bring me back to my feet. That is more natural. Okay, so that is a minimization process. You are able to bring the atoms back to their usual position, not usual, the energy minimized position, as it were. Okay, and sometimes some residues will be missing in the structure. So you need to add those residues. You are not the one doing it. The system will ask you that there are some residues missing. And you will guess which possible residues are there, and it will be added on and the rest of them. So the preparation takes place like that. The same way you can prepare your ligand after you either drawn the structure or you imported the SMILE structure or EMOS or SDF, whatever format. Sometimes you also need to correct the bonds and the charges. And sometimes you might have what we call the chiral or chiral centers that needs to be accounted for. The charges are very important in the sense that depending on where your macromolecule is situated, you may want to simulate at a particular pH. So what do I mean? There's a difference between the pH, intracellular pH of the physiological solution and the extracellular. The pH within a bacterial cell, different from the pH in humans. So these things are considered when you're designing a drug, because if you want the drug to ionize in a particular pH, you must have an idea of what the pKa of that drug is. When does it have a negative charge? When does it have a positive charge? And when does it have a neutral charge? Okay, so depending on that, you can add charges, and you can add charges based on the pH of the environment where you know the simulation should actually naturally take place. So you simulate that environment by changing the pH and adding the appropriate charge for that compound within that particular pH environment. Okay, and of course you minimize the structure, of course. Thereafter, for today, we're just going to see how one ligand interacts with one protein, but it's possible to take hundreds of ligands and say you want the computer to set up a system to see how each of them interact with one particular protein. That is very possible. So what we refer to that as docking a library of compounds against a particular protein. And you can also dock one compound against a library of proteins. So that is also very possible, okay, using some other platforms. All right. Some of the packages docking packages that are available is Autodock. We're going to use that within Chimera today. UCSF dock. And I also want to call your attention to some online services, especially if you're just using docking as part of, to answer a question as part of your main research. You don't need to install some of these things. You can actually do the docking online. So it accepts your receptor, it accepts a ligand that you want to test, and you tell it to do the docking, and gives it, it usually asks for your email, and they will give you an answer when the result is available. Then you can go in and analyze your result. But at least you need to understand the basis of what is going on. And that's the whole essence of this interaction today. Okay, so you can do it online. There are more than this, quite a number of them that you can. Haddock is another one. I think Haddock, I'm just going to add that. Haddock is a very good one also. Okay. The commercial ones are Glide Gold. These are more. Schrodinger is, we use Schrodinger here. And molecular operating environment, I think MOE, yeah. MOE is another good one. Most of the time I use this. These guys are also, they can give you a free license if you attend their webinars, and yeah, they give you a free license for 3 months for you to execute your project. So you can also check them out. Very easy to use and yeah, straightforward. Okay. All right, let's get to the exciting part now that we set the tone. Any question in terms of the principle behind the docking before we move on? Okay, I assume there are no questions if I don't see anything on the chat.
Yeah, and maybe before we go into the demo, our webmaster is already with us here so that we can take the group picture, then
so I stop sharing.
So yeah, maybe, yeah, because we have 20 people now, reduced to 19. We just have to take this. Then towards the end, if we have more number, we do it again. So, but that will require everybody to on his or her video. Then he will assemble everybody and take the video. The picture, yeah. So please, you are all required to now on your video for the group picture. And Edu, are you on? So you, on your video, on your chair, on your video. Okay, yeah. Bakare edit and Ray Izumo. Bakare and Ray, engineer Prof. Okay, ready? James or car for Ayale. Amara.
Hello, sir.
Yeah.
You will tell us when to smile, Abby, because I've been smiling since. Yeah, waste my smile.
Yeah, I think people are smiling now.
No, I don't.
Yeah, Innocent, because Aniedu is not yet here. Chikodle is off again. Azubike. So he is the one to join again. Sorry. Stop.
I think you can use screenshot.
Okay.
I will use snip tool. Just snip tool.
I'm using Mac, so alternate control.
So, sir, if you are taking a screenshot, so there's a Windows sign on
I
the
think
laptop.
I've already, I'm using another form of computer, so, but I've gotten it. Yeah.
Okay.
So, yeah.
All right.
I use that's how it goes here. But I think I've had one now. So maybe for the final, again, could you open the beautiful face again? Then I will do the final one now. Edit, you want this for. So who is not on? Thank you. Then you could come a little bit closer to your screen. Yeah. Okay, yeah. Joy, Giniga, what's happening? Chifu.
Can you see me now?
No. Okay, you have two connections, I see. Okay. 19, Chifu. Atanda, could you on your video for the last time? Sorry. I don't think Professor is connected. Yeah, 20. Okay. Not bad. Thank you. I think we can, Doc, you can move on. You are smiling too much, but it's okay. Yeah.
Okay, thank you.
Yeah. You can move on. Maybe if we had some.
Yeah, it was good to put some faces to the people I'm talking to.
Yeah.
Right?
Yeah. Yeah.
Okay. Thank you. Okay, I'll just switch up my.
Yeah. Okay.
Okay.
So
let's
we
get
have
our,
to go on.
let's get our camera on
again.
Yeah.
Okay. So you can off your camera to have some data. Thank you.
So let's go back. I'll share again. All right. So what we want to do is just to quickly dock one compound into this protein called plasmapsin 2 from Plasmodium falciparum. That's the PDB-ID. And the drug that I want, this is just random, okay? It might be in some cases like a couple of people that have done research that I've worked with on that side. I've looked at natural compounds that have been used ethnobotanically for malaria, and they know the compounds that are present or phytochemicals present there. So they want to know, okay, are those phytochemicals acting by binding to plasmapsin in order to bring about their mechanism of action? Okay, so that's one way you can kind of hypothesis you can draw, you can design. But this one I just picked this at random, this drug at random, not to this, it's binding to, sorry about that, 1LF3. Okay. So the drug I picked is an approved drug from Drug Bank. So I wanted us to look at Drug Bank today. We've seen PubChem, we've seen the other Chem EBI. All right. So I'll put in context what we want to do. Then I'll go to show you. So the first thing I want to do is to find this compound from the drug bank. So please flow along with me as much as you can. But I need to get this done quickly. And you can always go back to the video. I'll keep repeating myself so I want to go and look for this drug, which is going to be my ligand, and this protein is going to be my receptor. Those are the two things we need. This drug is going to be my ligand, and that's ILF3 is going to be my receptor. So I take that, and I go to my browser to look for drug bank. Okay, I've done it before, but it's fine. I'll do it all over again. I'll type drug bank. Drug bank. You can, I think it's
drug or drag.
Oh, it's drug.
Yeah.
My bad. Thank you for Google for correcting that. Okay, there we go. I'm in drug bank, and I can see it there. All right, good. And you click on that to get into the site. All right, so, and I just paste the name of the drug that I copied from my slide. I'll give you some time to copy that name, or I can just put it on the chat. Can I do that? Where's my chat? Okay, it's on the chat. If you want to copy from there and paste it there. Okay. So try and follow me as much as you can. Then just enter. So once again, now this drug bank is a collection of approved drugs, experimental drugs, or drugs that has been investigated. So there are drugs that have gone through some level of preclinical development. Some have gone through clinical trials and are approved. Some are still going through clinical trials, So that's the collection you have there. So I mean, like you have for PopChem, you have information about, for example, for this particular drug, information about the targets, the enzymes, the carriers, and the transporters. For those that are interested in pharmacokinetics, you know about carriers and transporters when you talk about efflux pumps and the rest of them. All right. Yeah. And target is the one that's your pharmacodynamics trying the particular target that it interacts with to bring about its action. All right. So you get all this information also for non-drugs. And what people are doing is that although this drug has been approved, for example, now is approved for it's a cardiovascular condition, I believe. I'm looking for the indication. It's indicated to reduce the risk of cardiovascular death, myocardial infarction, and stroke in patients with acute coronary syndrome or history of myocardial infarction. Okay. But I think I have this wider idea that this drug can also cure malaria. I have an hypothesis. Okay.
So in that case, that will be drug repurposing.
Repurposing. So it's basically called drug repurposing.
And that's a very good applications for us in the local environment.
Yeah. Because with drug repurposing, you don't go through the drug development again. It's already approved.
Yeah.
It's just an indications change that it might be required. People are saying ivermectin should be used for COVID. There's a lot of shout around that and everything. So that's the concept.
Do you have anybody working already on this drug? I mean, potentially for malaria?
No, this is just a random. I just picked it at random. Maybe it won't be the one I will pick because I won't pick a drug that is used for cardiovascular to treat malaria. Yeah, okay. I might begin to see because the current indication now becomes a side effect. The new indication.
Yeah.
So that's the bane of drug repurposing. You need to, yeah. Is there something on the chat for me? Okay. Okay. All right, so let's move on. So I need the structure of this. That's what I'm interested in. Remember, docking, we need structures to do docking simulation. So download, you look at different formats. More, remember the formats we mentioned. We just check them out, please, if you need more information. MOL, SDF, 3D SDF, PDB, SMILES, and the INCHI key. But I want the SMILES. Okay. So that's what I did now. I'll go back again and repeat that so that you can always do that again. I click on SMILES and it took me to a new page where I can copy the SMILES. So I'll just go there and copy that SMILE. All right. Then I'll go and put it somewhere. I think I've done that before. Yeah, I just put it here. So that's the first SMILE format for that drug. Okay. So the next thing I want to do is to find that protein. Okay. So I go back. Now we're engaging with our biological database now. I go back to, so we're done with this chemical database. We want to talk about, we want to go to PDB. And the code for that is ILF3. So I'm going to search for it. Did I say I or one? It's 1LF3. 1LF3. Okay. Crystal structure of plasmapcine from, okay now. From falciparum, plasmodium falciparum in complex with an inhibitor. So this has been co-crystallized with a particular compound, which they called EH58. So these guys that did this, it's a novel compound that has not been made before. Now, because they've co-crystallized it, we know that this particular protein, we have an idea of the binding site or the active site, because this was designed to bind to the active site and then now co-crystallize it. So very good, we have an idea. Sometimes you don't know, like we said, you have to find it. Okay, so very good one to use. Then you can go and download it, the PDB format. Then you save it. I've already done that, so I'm not going to say it okay. Say okay. So you can do okay and you save it. Okay. Now I want to give you just 1 minute. I want you to create a folder to store all things, please. All the things we're going to work on. Create a folder now and store them in. So what you just downloaded should be there. And every file that we're going to talk about now should be there also. Another thing that I forgot to mention at the beginning, sir, we should actually, if I get an indication from people that they have created the folder, I want to move on to something that I want them to.
Okay.
But it's a prerequisite to this.
So please let us know in the chat.
Let us know if you have moved the folder so that I can give you the next task.
Yeah, so once you create the folder, you apply what you have learned in the last 2 days and get some information.
Okay, so now look at this. We need to download Autodoc Vina.
But you wanted them to use the one already built in Chimera.
Yeah, but built in, but it's not, you need the executable file.
Okay.
You need the executable file. It's a simple, it's not serious installation. Okay, can everybody do that, please? And as you do that, I just want a response. Done, wait for me, or whatever. So you see Autodoc Vina in your Google tab, then you go into the download section. And you'll see different formats for download. Windows,
Mac.
Linux, Mac. Yeah, you see them. Then you choose the appropriate one for you. So I'll choose Windows. I'm using the Windows system now. And you save it. Are we at that level? What did you do? Thomason. Where's my tab? Beautiful. Okay, not following.
Hi.
Go back to Google. And do I need to type the steps? Type
Okay.
Autodoc
All right.
Vina in your Google. You want to download, type that. Once you have that, then you click on that Autodoc Vina molecular docking virtual screening tab that comes out. It should take you to the page. Then you click on the download tab on the page. The Autodoc Vina page. Then you should have something that looks like this that I'm showing. Thereafter, you need, if you're using a Windows system, click on the one that is in front of the Windows, and it will ask you the place to save it. And you just save it. All right, cool. Okay, can I get a feedback where people are? So you should have your installer in the folder. I have mine in the folder. Oh, some people are installing already? Okay, beautiful. Okay. Okay, so let me just do this. So some people are ahead of me already, so it's fine. That's good. Install. Now, in this case, because I've installed it before, I'm just going to remove it and repeat the process so that you can have it on record. Hi, Bo. Okay, so this is, if you are installing from the fresh, this is what you do next. You want to read the terms and conditions, you can read it. Then you agree with it.
Definitely must agree.
if you want the thing. Next. So for those that have installed it, good for you. For those that have not, you can change where it should be installed at this point. So if you have installed it already, you need to know where it is because you're going to need it. Usually it goes to the program and it creates a folder for itself called the Scripps Research Institute and creates another folder called Vina. Then it puts it there. Okay, this is what it does. It goes to your C drive under your program files. If you're using Windows x86, He creates a folder called Scripps Research Institute, another folder called VINA, and he posts the executable file there. Okay. But I can change it if I want to. I can put it somewhere else. So just a next, and it does its job. Allow it to do its job, and it is installed. Okay, cool. Still. Okay. Are we done? If you're done, I want you to find the folder where you installed, where the thing is installed, and copy the VINA exec file. And paste it in your working folder or what we call your working directory. I'll say that again, and I'm typing it. So if you have installed it, you need to open your folder to find, you need to find it now. So you go to your C drive, for those that just didn't change anything, your C drive, your program times x86, it should have something called the Scripps Institute folder, which is that. I open it. There's another folder called VINA. I open that. And inside that folder, there's a VINA application. There's a VINA split, there's a VINA application. So I'm not interested in the split, I'm just interested in the VINA. So copy this. Then you go to where you are working. I have a folder called, on my desktop called Docking Unizik. So I paste that VINA in that folder. Just paste it there. Cool. Now I want everybody to get up to speed to that. Everyone, please. So if you're done with it, just type done. So I'm expecting 21 done.
Yeah.
So I understand what I said, Abi. So at least
Yeah.
you can repeat to them.
There should be 21 done. Yeah.
I just pasted it. When you see the VINA, copy it and go and put it in your folder. Don't click on the VINA. Don't try to open it. Don't just copy it and go and paste it in the folder that I said you should create for this work. The VINA where your protein is right now. Don't try to double click it. Don't open it. It's not gonna work. It doesn't work that way. Okay, I'm still waiting. Doc, let me just get the garage for my wife.
Okay.
I'm waiting for the done, please. Yeah, just wait. If you're done pasting, just wait, please. so that everybody was on the same page.
Yeah, we need 21 persons to have done.
Hello. Oh, I said don't open the file. You're just Once you install it, just copy the VNA and just put it in your working directory. Okay, done. Okay, some people were not.
Yeah, sorry, some persons missed out.
Were discarded. Did you? Okay, are they been intimated? Okay, do I need to paste this instruction again? Downloaded your document from Google's. Or is it your network? Okay, once you have your form downloaded, you have to install it. If you're at the point of downloaded, you have your folder that in your whatever it is downloaded into that looks like that. Okay, let me make this bigger. If you look at my screen, let me try and make it bigger. No, I can't. So you have something downloaded called Autodoc VNA 112 Wayne 32. If you just downloaded it, then you need to install that. You need to say right click, install, then you go through the process of next, agree, next. The same way you install most Windows installation packages. Okay, some people are already done. All right. And for those that are if you just install, you need to go and find it. And we told you how to find it. So I'm just going to do this one more time because we need to move on. It should be your C drive. You can always go back and check this. In your C drive, your program files times 86. If you didn't change anything during the installation, then it should be in your Scripps Research Institute, the guys that made Autodoc VNA, then you should have something like this. Then I want you to copy this. Just right click, copy, don't open. Copy, go to your training folder or the docking folder, whatever is the one you have chosen. My one is docking Unizik, and I right click here and I paste it in there. That's all. Done. Beautiful. Okay, so I need to move on. So can I move on? I don't have 21, but I think I need to. Okay, where are we? You click on the download. Sorry, are you still downloading? Please, Lillian, can you unmute? I need to understand where you are.
Just make that because I cannot do that.
Okay. Okay, if the tutors can reach to Lillian in terms of maybe, I don't know whether it's easier to reach by phone. Just right click on it and say install. You have to double click or right click on install. Yeah, if you double click on it and say install. Okay, let me repeat that again. And I want you to follow me as I do it, please. It's just that I've installed this like 100 times. Okay, I'll say install. Now mine is giving me this because I've already done it, so I'm going to remove it again. So that I can reinstall it. So just give me a minute. Okay, so if you do right click install, you will get to this page. Okay, then you say next. You agree with the terms and conditions. You go next. Then you will see where it will be installed here. I just want you to take note of that. It's in my C drive, it's in this folder, that folder. You know, I say next, next, and it will be installed. You just need to confirm, that's all. So once you're done with that, whoever else is watching, you need to go and find it, like I said. So you go to your C drive, programs, the Scripps Institute, VINA, that's VINA, not the VINA split, VINA. Don't open, don't write, don't run, just copy and go and paste it in your folder. All right, in the folder that I said you should make. Okay, so let's move on once you're done with that. Okay, I took a detour from somewhere. Do we all have the smiles copied from drug bank for the drug? I'm looking for my own drug bank. Yes, okay. Done, okay, beautiful. So Lillian, you find it, copy it, and paste it. I will go back to it again. So you can just, I think you should just follow us now. Before we do the execution, I'll go back to it. All right. Okay, so you have that smiles copied. I just confirmed from a couple of people that you have the smiles. This is mine, copied somewhere. All right. So from this point, we are going to Chimera. So I'm restarting my own Chimera. It's been up since morning, so. You paste it, I said you paste it where the folder that I said you should create. Lillian, you paste it in the folder that we said you should create. I believe you have downloaded your PDB, Prot receptor, so it should be in that same folder. But like I said, you can leave that for now and follow us with this. We'll come back to that just before we run the simulation. So everybody gets your camera fired up, and you should be on this fresh page. If you are working on it before, you can close that session and start again. So I need you to copy that smiles that we pasted somewhere. We need to copy it because we now want to build the ligand model and prepare it for docking. Okay, so I went to pick the, what do you call this? Oh, I hope I've not messed up my mouse. Okay, I'm going to take my mouse, copy. Then I go to my camera that is open. I go to tools. Where am I? Structure editing. Then I want to go to build structure. So I'll repeat that. I've already copied the smiles. I go to tools, structure editing, build structure. One more time. Tools, structure editing, build structure, and she'll open something like that for you. I'm trying to bring it to the center. This is where you build your initial structure. Now, there are different options. You can build from fragments, from atoms. You can actually also fetch the compound from PubChem. So we can technically actually look for that same drug in PubChem, get the PubChem ID and CID and put it here, and it will fetch it. But since we have the SMILE string, we're going to use the option of SMILE strings. So under that build structure interface, select SMILE strings. And in front of it, you'll see SMILE strings and paste it there. So you need to use Ctrl+V to paste it. It doesn't allow right clicking. It's a Python. So Ctrl+V to paste it there. Careful that you pasted the correct one. You don't double paste. And you can change the name. The default name is NUK, but I can change it to, okay, I don't want to call, let me call it something else. I'll call it Uni. It doesn't allow more than 4 characters. Okay, the residue name does not allow more than 4 characters. So please take note of that. So you can, once again, you can paste it here, the SMILE strings, and you can change the name. Once you're done, you just click apply and tell me what you see. Beautiful. Oh, you are looking for the smiles that I copied. Let me put it up again on the, oh, that's directed to you, Lillian, to everyone. What is going on? Okay, so do I assume that you didn't follow what I said? You just did. Or somebody else saw you seen a structure? Okay, I've also seen a structure. Okay, you just wait for the rest to see a structure. If you have not seen a structure, you go to Tools, Structure Editing, Build Structure. You paste your stuff in that space. You can change the name, something that you can remember. Then make sure you have selected SMILE string. Then yeah, you should have something like this. Beautiful. All right, now we have the structure. So what is the structure that we have? If you go back to our slide, that is the one we downloaded from DrugBank, right? that is this drug, ticagrelol. That's what we downloaded. So that's the structure we have that we're seeing right there. So we need to prepare this structure. And in the preparation process that I spoke about, adding charges, adding hydrogen, minimizing the structure so that it gets to the conformation that allows at the minimal energy. Okay, so let's do the minimization. I'm just going to wait a few seconds for people to get to the same page. Beautiful. So we want to prepare. So most of the things are under tools. You go back to tools again, surface binding and analysis. And under that, you'll see doc prep. Doc prep. Okay. So once again, let me do that again. Tools, surface binding and analysis, doc prep. I'll leave that on for a few seconds. Tools, surface and binding analysis. That should open another tab. Then you can select doc prep. So if you click on your doc prep, it will open another interface, or what do you call it now? A dialog box. So, and if you just take a look at that, you will see for this chosen molecule, do the following: delete solvents, delete non-complex ions. It's looking for alternate locations, keep highest occupancy, stereoisomers, especially when you're using minimizing protein. And yeah, just a minute, please. Okay, sorry about that. I'm back. So I click on that, add hydrogen, click on that. So it just brings in more dialog box as you go step by step. Click on that. It gives a neutral charge. Remember what I said about charges. If you have any information about the environment you want to simulate, and you feel this shouldn't have a zero charge, you should have a +1 or minus whatever, you can select the charges here. But yeah, I don't have any information about the environment, so I'm keeping it as a neutral charge. Okay. Then it brings me to the point that I can now save the prepared ligand. So I started with a particular ligand that I put in built structures. Now that I've gone through those processes, I can now save this ligand. So I'm going to repeat that. Yeah, let me check if there's a question. Okay, I'll do that. Okay. Tools, structure edit. No, it's tools, surface, and binding analysis, doc prep. So delete solvents and some other processes that it will do. If there are incomplete side chains, this does not apply to this small molecule. It applies to proteins. Add hydrogen, add charges. Then the final thing, it will write a Mol2 file. Once it's done with all this process, it will write a file that we need to save in the same folder, please. In the same folder where your PDB file is, in the same folder where your VINA application is. Okay, so click on that, click OK. Click OK. Click OK, step by step. And I spoke about the charges if you want to just. So for now, let's all use zero charge. So when you do your own thing, you can do that. But there's an option to change the charge. And click OK. And once it gets to the last step, it's going to ask you, where do you want me to save this? So you need to give a name. This is my test drug 1. I'll call it that I just prepared, and it's going to save it as a Mol2 file. And I'll say save. Okay. So once you've done that, confirm that you have that in your folder. So I can go back to my Unizik folder and look for test drug 1. There it is. I can find it there. I'm happy with that. I'll give a few seconds for people to come up to speed. So somebody is saying they can't see me, the screen. It might be from your end. I'm still sharing my screen. No. You're not done yet. It's still processing. It's still processing one of the steps. If it's a black screen that is blinking, don't do anything with it. Depending on how fast it will execute some of those processes, depending on how fast your computer is. Just wait for that screen to disappear and you will see another thing. Okay. So I just said, if you see a light on screen, it means that the process is still running. Just wait for a bit. You should finish and clear that screen. Then you will see the next dialog box. So if you have succeeded in getting to save, or you have saved, let me know. I need to repeat the process. Okay. Okay, somebody has saved successfully. Okay, can I get 2 more people? We have. I still have 1. Yeah, the station is .mo25. Yes, thank you, sir. Do I have anybody saved already? Any other person? Because I want some. If there are challenges, let me know, please. Just type it if there are challenges. How do you save? Just a minute. Tommy, just give me a minute. All right. You still didn't get a saving prompt. They repeat the process if you didn't get a saving prompt, attender. So it's another person has saved. Oh, then your system is slow. The system is slow. If you didn't change anything, you just go to, I've repeated it. Another person saved. Doc prep. If you didn't change, okay. It's almost automatic. It should be okay. I mean, it might, well, depend on the system you're using. It's fast enough. It doesn't show the blank page. Okay. Okay, to the next. That's blank page, very brief. And it brings me to this saving thingy. Yeah, this one. And I can type the name here. After I've navigated to the, remember you need to navigate to the folder. Okay, then you, yeah, type the name, save. All right. A couple of more people have saved, yeah. So if you have saved, let's repeat the same process for the, we're going to repeat the same process for the protein. If you have saved, so if you have saved, go to File, Open, navigate to where your protein is, your PDB file that we downloaded. That was there. 1LF3. You know, then you can do the same thing and save the .mo2 file again for that protein. I'm going to do that in a minute. And just perhaps before you do that, you need to get this one out of the way. So once it's saved, I can just say select and delete it. I don't want it to be in the way for the protein setup. Okay, remember to delete. You select it, action, atoms, delete. So please delete what is left because you have saved the one you want, which is the mo file for that ligand. So you can delete it so before you bring in the protein. I didn't download the protein, so I'm going to fetch it, which is another option. That's a 1LF3. I'm just going to fetch it straight from there. Okay, I have it here. If you have your protein, please do the same quickly. Tools, structure, surface and binding, doc prep. And it should follow the same process, except there are some special things there. Prep. So for those that have slow system, at times are you able to save? It has, okay, eventually finished. The protein might even take longer. So I don't know. Okay. At the bottom, you will see the things going on. It's asking for the charges for the ligand that was bound to it. I just keep it at 0. I'm not even interested in the ligand for now, because this is the blank page you guys were talking about. It's done that and brought me to the save page. So this is my receptor. I'm going to call it receptor. I'll call this one receptor 4. I think I have a couple of them. Okay, and I save that. Okay, so I'm going to repeat it again. So 2 options to bring in your protein. You open, you go to where you downloaded it, and just click on it. Is there a question?
What is the protein we have to fetch?
No, did you download, Joy, did you download the protein earlier? I didn't download it, I was just showing you. Then, so if you downloaded it, you just fetch. If you didn't download it, you can fetch it. Let me write it for you. If you didn't download it, that's the protein. Okay, you selected, you don't know how to delete. Let me do that again. Once you select something, I don't want to delete this protein anyway, but assuming that, then you go to atoms, under actions, atoms and bonds, and go down to delete. I know it takes some getting used to. As you keep using it, you remember where these things are. Good. So have you saved it? Vivian, have you saved it? Have you doc prep it? Okay. Oh, we just deleted. So you need to bring in your protein. Okay, cool. Okay, so bring in your protein, you can do that. Open, bring in that, or you fetch it by typing 1LF3, and you fetch it. Once you're done with that, do the same thing we did for the ligand. Doc prep. And you don't change any setting. Just keep saying, okay, 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 I agree with everything you're doing at this point.
Yeah, at this point, yeah.
Okay. But when you repeat this on your own, please take note of the steps you are taking. Because when you're writing your method, these are the things that you need to write in your method. Okay. Sign, for example, I can say charges were assigned using this force field, amber FF14SB. You need to specify because there are other force fields that are present here, and the result will be different. Okay, so remember when you're doing it on, you need to take note of all these steps that you took and the specific parameters that you used, the ones that you clicked, the ones that was highlighted when you're writing your method. Okay, this is okay. It's done that. Assign charges to the ligand that is there. It's done that and asking me to save now. Then I locate my folder and I save it. Okay, tools, running analysis, doc prep. To delete, you just select it, select. Remember, you deleted yesterday, select chain, you select you will see instead of what I have here, you will have ID. Just select that ID. Then you go to actions, atoms and bonds. Then you at the bottom, it should be delete. That's for joy, I guess. No, you're not doing any structure editing. You're just selecting. Once you have the protein, you go to tools, surface analysis and binding, then you doc prep. You're not doing anything in structure editing. Hello. Yeah, the protein already has a structure, but the compound also has a structure. But the format that we took from the drug man is a SMILES format, which is a string. Okay. If you put that, if you can't just put that string in like that, you need the computer to edit that string and produce a structure that you can see like this. So that's why we went through structure editing. Remember, I said that at that point, yeah. Okay, I'll get back to you now. Let me just finish this. Why we didn't do structure editing? If you do that to structure edit thing, build structure, you can actually go to PubChem and type in an ID. And if you do that and apply, what you will get is a full structure, just like you did not, you got for protein. Okay, so that's the reason. Okay, please, what should we do with the dialog box that comes up when we get to doc prep? Just click away, click OK away, please. Okay. Okay, so this is the last bit of the thing where it comes on the home run. Children have put the pressure on it. Okay, do we have anybody that has saved the protein?
In what format did you save?
What do you mean your protein is Bankole? What do you mean still running? I don't understand. You mean the preparation is still running? No, the protein will be saved in multiple format. It's default. Okay, thank you, Mr. Ike. Thank you. I believe that's, yeah, it's a mistake. Okay, sir. Saved. Another person saved. Good. I'm also saved. Okay. Thank you, Amara. I agree with you. Yeah. You can feel my pain, Amara. My children are already pressuring. Let's go. Okay. All right. I'm glad we got this far. At least one more thing that I want us to do that might be a bit involving, and the rest should be just be clicking and clicking. All right. Okay. Now, I just want you to listen to this. If you are still working, just listen to this. You would definitely get the recording, I believe.
Of course.
Yeah.
They had some. I've not even gone through the recording of the other days. You will get it. It's not a problem.
You will get it. Okay. So
remember to share with your sound where you want to share the voice.
You said?
You want to share a voice note?
No, it's fine. I'm talking.
Okay.
Okay. So what do I want to say now? So we have, for those that have gone to step with me, you have your ligand saved, you have your receptor saved in your folder. Okay. So I can actually delete this one because I have the one that I want saved. So I can go to select, chain, action, atoms and bond, delete. Good. Now I'm going to go to my own folder and bring in two things that I need. Okay, two things that I need to execute this process. And the two things are the receptor that I saved. Mine was receptor 4. In MOL format. And the ligand that I saved, which I called test drug 2. Good. So I have the two things I need to do the docking. This is the ligand that I want to dock. This is the protein. And what I have inside here is the ligand that came with the protein, the co-crystallized ligand. Okay, that's what you have inside it. Please follow me and just watch this last bits. Then you can go back and try it. Now, like we said, most of the time we usually define the binding site so that the algorithm know where to place the ligand. Okay. Now because this has a co-crystallized ligand, it gives us an idea where the binding site is. So we can say, okay, fine, this is where we want to dock. So we need to tell the algorithm the dimension and the direction in space where it should do the docking. Okay. The direction and the dimension in space, what we call a grid, a search space, okay, for it to do that docking. I said that again. We need to give it the dimension and the direction where it needs to do that docking. It's called a grid box or a search space to do that. And because we have a co-crystallized ligand, we can use this as a guide to design our grid box or the search space. So to do that, I have to go to tools again, surface and binding analysis. Then I call up my Autodoc VNet that is in here. Let me move this things. Okay. The Autodoc VNet toolbox. I will repeat that for clarity. Tools, surface and binding analysis, Autodoc VNet. So it comes with a dialog box. Okay. So the first thing on that dialog box is output file. Where do you want me to put the files that I generate from this process? It's asking me. That's my output file. So I need to browse, and I browse to the same folder that I've been using. Okay, and I'll go there and I say the same docking uniseq puts this process of, let me call it my plasmepsin test doc 1. put it there. You just click on set output location. Okay. Now, what are you docking? I'm docking a receptor called ILF3. Which one is your ligand? It's asking me. And I said, this is my ligand. Remember, we brought in the smiles as the ligand, and that's the blue one. For you to confirm that, if you go back here and say chain, you're going to see this, which is, if you select that, that is that one. If you go back to select again, chain A, and select smiles, that is this one. Remember the ligand you want to dock. Okay. So when you get to this box, you can select the appropriate model. This is the model that is the receptor. This is the model that is the ligand. Now, we need to now set our search space so like I said, the direction, which is given by the coordinates, and the dimension. So the direction is established by the coordinates. The coordinates is a set of X, Y, Z in space. All right. Now, I don't know what the coordinate is in this protein, for example.
I think it's like 3-4 pieces video.
So what I can do is to
I send you
go big again.
also with
Then I'll say -5.
Okay.
Just again.
30.
this is 30. I mentioned this.
Hello?
Can you mute so that
okay, yeah.
Okay, thank you, sir. All right, so I chose a random coordinate. Then I choose a size. I want it to be equal side. Let me choose 10 Armstrong, 10 Armstrongs, length, breadth, and height. Okay, once that is done, what you will see immediately I finished inputting all the values here. It's a box, it's a green box. You see a green box here, but it's not in the correct location. I want it to be around this ligand, which is my binding space. I want it here. So somehow I need to move this box to this particular location. So to move the box, just pay attention to this. Like I said, don't do anything, just watch for now. I need to click on this resize search volume using button. Now I got a series of buttons here. I'll choose button 1. Button 1 allows you to move the box. Button 2 allows you to move the whole thing. I'll say that again. Button 1 allows you to move only the box. Button 2 allows you to move the whole model. Okay, so this is what I want to do. I want to move the box now. So I say button 1, and I put my cursor not on the box, anywhere near the box, and I can drag the box to a particular position. Now, when I want to now view if I'm in the right position, that means I need to turn this whole model. I need to go back and change to button 2. Then I move the whole thing to find out where I am in my attempt to put a box around that co-crystallized ligand. All right. Now I'm not there. As I moved it, something has changed here. The coordinate has changed. Okay, you will notice it has changed. All right. So I'm almost there. I think I'm almost around that. But the box is too small because part of the ligand is still outside that box. So I can also change the size of the box. So I go back to button 1. So change the size of the box. I need to put my cursor on the side that I want to change. You can actually expand the box. So if I put my cursor on this side, I can expand this side. If I put it on this side, I can expand this side. If I put it here, I can expand this side. So it depends on which side I want to expand. Then I can go back to button 2. I can change it again, find that, okay, is it covering everything? Okay, not yet. I can see on this top, it's still not covering everything. And it's slightly at the bottom. Okay, so can I adjust the bottom first? I go back to button 1. I put my cursor there, I pull down a bit. I go back to button 2, I reorient myself. I say, okay, the top is still exposed. I'm going to change button 1 again, and I pull up that box dimension there. Then button 2 again to move around to see where I am. So I've done fairly well here. I tried to cover that. You can do better than that, I believe. If you feel it's too big, you can adjust. If you feel you need to move it, remember, all you have to do to move it, if you go back to button 1, is to just be on the outside. Oh, I've changed my thing. Okay. Okay. Okay, now my thing is too big. I need to reduce it. All right. So I'm just going to use this one. I'm not going to get too finicky with this. All right, cool. So that's my grid box. So what you need to do is to copy this and write it somewhere. Because anytime you want to do anything on this protein again, you just need to enter these parameters and you can have the dimension and the coordinates of your grid box. So for me, I'm just going to snip it in case it disappears. I'm going to snip it out so I can paste it somewhere. Okay, I'll snip it, I'll copy it, I'll put it on my slide so that I'll remember it. Yeah, there we go. That's my grid dimension. I'll keep that there in case I need it again. All right, so we've done the output, we've done selected our receptor, we selected our ligand, we've defined our search space. The rest of the options are default settings. I'm not going to change anything with the receptor option. The ligand options, I'm not changing. Advanced options, the number of binding modes that you want it to give you. The more binding modes you get, the more options in terms of possible position that ligand can take. But analysis becomes a bit more confusing because you have a larger number of poses. You need to decide which of these poses is most likely, but you have a more exhaustive research, but at least data, but you have to do more analysis. Exhaustive search is basically saying, do as much as you can to find the right position when you're placing that ligand into that space or that binding site. And of course, there's maximum energy difference between poses. That is that. Then executable location. Now, this is where I wanted you to put your VENA in that folder. Okay, so there are two options to execute this. You can execute this on the web service provided by the guys that operate Autodoc VENA, the Scripps Institute. They have a server. So if you select that Opal web service and just click OK, all the docking process will take place on their server, not on your computer. Okay, so if you click the first one, like I said, it has a server ID, which is put here. Please don't reset it, except you can remember it. Okay, it goes to their server. Except you have your own server that is created for this, then you can reset the server. Then it connects to your server and runs the job. Okay, but you also have an instance of VENA on your local computer, the one we downloaded earlier. So if I say local, I can browse to where my VENA is. And that's where I wanted you to store it, in the same folder. Okay, I can browse to where the VENA is and I just go vena.exec. That's the application that I put in that folder. And I click OK. So that's the path to my VINA set out there. Okay, so everything is taken care of. Your output folder is defined, your receptor is defined, your ligand is also defined. What else? The search space is defined. We're leaving the options the same. And of course, your executable file, where the VINA will execute it on your local machine, is also there. So just click OK. If the stuff is good enough, it should run for some time. So it's saying running, you look at the bottom, it's saying running at the bottom here. So it's doing its job, but I'm not going to wait for that for it to run. So I will have to skip this process.
Doc, yeah, so I appreciate what's going on now, and I'm just thinking around, for example, where the presence of the crystallized ligand, do you think this will affect the docking? Maybe already block the site you wanted to because already the ligand is there, or should anything?
Yeah, it does not. Yeah, it actually ignores it.
Okay, perfect.
Yeah,
but that gives us an idea.
It just gives us an idea where to.
So what of a situation where we could have another probable better binding site away from where this ligand is showing us in the protein?
As long as you know it, you can just, once you start with a random coordinate and you get the box, you can drag that box to wherever you want it to be.
Okay. Of course, so yeah.
One of the easiest way to check is, for example, for now, if I say, select this chain now, this protein, and make it a surface. Show the surface. If it can do the process all at the same time. If it shows the surface, you will see why that place is the binding site. Is the main binding site. But you can also see some other binding sites around. Okay, now I'm overloading my CPU.
Yeah, I see. It needs messy. Maybe I'm still narrowing to, for example, I have several natural products, and I'm still interested in my resistant malaria studies, and we expose all of them to this. We'd all be competing for one particular binding site, or we'll look at, the several binding sites, and now from the binding affinity, decide what happens, and they want to continue with maybe in vitro, and move on from there.
Yeah. So so you can definitely explore multiple binding sites, but we all know in reality the ligand will go to. We engage with the protein at all sites, all possible sites.
Yeah.
Okay, cool. I think I'm going to terminate one of this process.
Which one are you waiting for? You are responding to the initial question I asked with the looking for the surface. Okay, I can't click your own stuff. So,
yeah, that's what I'm doing. But I think the process arrested my CPU.
Yeah, maybe you could stop that one so that we wait to conclude the trading first.
Line. Hello, Doc.
Yeah.
I will have to shut down and come back. I think this thing is not, it's frozen. So when you're doing this docking, you have to be careful not to do some other things. Maybe it's better to do it on their server. Okay, I'm back, but my camera is shut down. It's fine. So I'm restarting my camera. So because I've done this, I think I did it twice today. So I'm just going to show you what the result looks like at the end of that run. It's going to put some files in your folder. Okay. So the ones I did earlier was plasmepsin doc test 1. So a series of files that will be in your folder was called a PDBQT files. PDBQT files. Highlighting a couple of them now. Plasmepsin test. Okay. So this one, especially the one with the higher size, is storing my what do you call it? My conformation that I made, the poses from the I can, let me just finish what I'm doing and I'll attend to your question. All right. So like I said, I've done this before. So I'm going to go back and open my receptor 4. Because I want to see the output. So if I go back and I open my receptor 4, what I docked again. Okay. Then I'm going to open. Now this I want you to please pay attention to this, because once you finish the post, it's going to store them in your folder, and you're going to have a PDBQT file. Bros, can you mute? These guys are talking. Doc, thank you, sir. You're going to have a PDBQT file in your folder. So you can't open, if you click on this, it's going to start telling you stories or give you a text file. We need to open this file in the Chimera. So to open it, you don't go to open, you need to go to Tools, Structure Editing, View Doc. You want to view the result of your docking. Okay, I'm going to do that again. Tools, Surface and Binding Analysis tab. View your doc result. Then you go to look for that PBDQT file that I just identified now. What was the name again? PBDQT test. I hope it's the test. Yeah, test 1. Okay, that's not the one. So which one is my result? PBDQT test 1 ligand. I'll do that again. Tools, View doc. Okay, so where's my ligand? I didn't wait for this one to finish, but I guess if yours finish, actually, when it finishes, you should get the result straight onto your visualizer. But now I didn't wait for that, but I'm looking for the one that I've done before, so I can show you a sample of what it will actually look like. Not this one. Which one was it now? Okay, let me check the test file. No, leave it. Okay, one of them is it. I will check. I'll keep checking. I have so many of them, like 3 today. No, it's not this one either. Autodoc. No, that's not that one. It's the ligand. So no, I'm dealing with so many files here. It should be easier for you because you shouldn't have so many files in it. Just the one you just. You're still struggling. I don't want to repeat this process. Oh, shit. Okay, just for the purpose of recording, I'll just leave it.
The one you are running, is it done?
No, you shut down.
Oh, I
because
see.
my system, it shut down. Okay, I'll repeat it. I still have the grid, so I'll just.
Yeah.
I'll repeat it.
Or we might give you space to restart the entire system.
No, it's fine.
Okay.
I'll repeat it. What did I call that one? Let me call another name. Plasmid C doc test. I don't even remember that. Test 2, test 3. Let me call this test 3. Set output location. Receptor is that. Oh my word. Okay, that's receptor. Okay, which one of them is my ligand now? To confirm which of them is my ligand. Change. Okay, so. Okay, so 2 is my ligand. All right, I need to remember to fetch the
Du weißt nicht zufällig Benjamins Geschäftstelefonnummer auswendig, weil ich muss ihn mal schicken und TNT.
I'm gonna be lazy now. Just doesn't make 30. 11. 16.
Ja, gut, das muss auch funktionieren.
Und ist ja schon auf der Homepage da?
Ja, ich habe geschaut, aber nur auch keine E-Mail. Also E-Mail hatte ich, aber auch keine Telefonnummer.
Ein Foto schon? Na gut, also ich kann dir gerne schicken. Willst du es mir ein bisschen?
Nee, lass es, weil Tatjana hat ihm jetzt auch mal geschrieben. Auf die Telefonnummer, das passt schon.
Nicht schlimm.
So it's running now.
Dann kann ich das Chatline
oder Pop.
So, vor die Tür hinein, aber ist ja normal.
Ja, nee, das ist ja nur, dass die, wenn das Paket angeliefert wird.
Sorry. Thank you. So I'm going to leave it for the purpose of record so that the result comes out after this, so that I can show you the analysis. Okay, so I'll just leave that on until it finishes. Then I'll come back and do the analysis.
Maybe we could also use the time to respond to the questions here. Hello?
Which questions? Are there questions?
I think just one.
Okay, cool. I'm trying to find it.
Okay, someone who is asking, Amara is asking how to locate a novel binding site.
What do you mean by novel binding site?
There's nothing like novel binding site, but maybe if already a drug is failing due to resistance, so another site for the drug could be novel anyway, because the previous ones that are known is already failing. So that is also the trial and the error around it. So is to find another site, but these should also be druggable sites, and also to know whether the site is quite essential to the organism or things like that, or if you drug the site, the organism will find another way to make switch through another part to go on with his life and all of that. So I think there are a lot about the sites, which, just as you said, there are websites that have done a lot of sites. Yeah, one could use. So it may not be novel because it's there, but it's just that you want to try this site for because already the other site has been compromised due to mutation or and all of that.
So at the correct time, I think, is an allosteric site. So you could find another site and say you modulate that site. As long as if you modulate that, it changes, it has an effect, like you said, on the protein, then good for you, you found something new. Although the site has been there, like you said, but it just shows that if you do something to another site, but most of the time, if it's a multi-domain protein, you do something to one site, it will not affect the other.
How many people are here? 20? Is Onoche here? Nochina is driving car and also doing bioinformatics at the same time. Yeah, it's usually difficult.
Let me show them. Let me show them.
Ich muss gerade nichts im Labor machen. Dann arbeite ich lieber von zu Hause. Außer, dass ich mich jetzt gekreuzt bin. Aber wenn ich nichts zu machen, ist mich ja auch trocken.
Das stimmt.
So maybe you could, I don't know whether it's like Prof was not when we took and Onoche, the other group photograph. So maybe we could do that again now. We still have 20 people. It's reasonable. So in that case, maybe Doc have to stop sharing his screen. Doc is under. Yeah, you can stop so that it will run at the background. Then we take our group photograph again.
Okay.
So View full screen. So you can on your pictures. Prof, we have taken already, but you are not on board. Then,
sorry.
Yeah. So let's once again on our pictures for the purpose of those that missed the other one, and they could take. You know, Sir Benjamin, your stuff is so bad. I don't know why. Yeah. But at this point now, yeah. Okay, Naomi, you are still around. You didn't go again. I thought you have gone for school run. Many people are not on. Maybe I should just take for so that Prof will see his own face. So we have many versions because people are even inside the car doing the bioinformatics. This is serious. Hey, my people. Okay, so
I'm sorry, I had already entered my vehicle to go for school run.
It's okay. 1, 2,
so
3,
that's
4,
why I'm in the
5,
car.
10.
All right,
So
thank
we're
you.
21 here. Chukwudi, at least you can snap picture today. Because, all right, you know, sir, Benedicta, Chifo, and Bakare. So finally, I go. Are you smiling? What's your plan? Okay, fine, nice. Done. Yeah, did anybody join again? I think it's oh no, cheer now. Okay, yeah, it's okay. Yeah, perfect. Yeah.
Thank you.
Yeah, but Edict Bakare, I think I took your screen, whatever. That is better also. Yeah. So we begin to round up while we wait for the run, because this run that is going on is very important. You have not docked until you get to this run. Yeah, so that you can be able to see what is the binding affinity of the protein and also the ligand, you know, and you could interpret, even though we're using only one ligand. So a situation where you have many ligands. So this is at the point to screen out some ligands. When you discover that the particular compound has a good binding affinity above others, then you can do away with the rest and continue your work with one into molecular dynamics. And remember, we say we have to at least give a definition to molecular dynamics so that in the course of the year, when we shall spend 3 days again on dynamics, to understand what is doing, what is happening between the protein and also the ligand in space properly. So yeah, so that is why it's important to round up this run, and from there we could begin to, then we're going to put together our assignments, and these assignments, well, I will send it to your email, then you do it in details, then there's a deadline to this assignment. Then upon submission of your assignment, you will receive your certificate of attendance. Yeah, if you don't submit, no attendance, no certificate. So in that case, because you can always go to Business Center to design a certificate for yourself. Yeah, but but for this project, but for this our training, then you have to do the assignment and send it in on a particular deadline. If you send it after the deadline, we might not also give you, but you have to do and send it back to us. Then, well, yeah, just do your best. The training should go past through you. I think that's the whole thing. So for those who miss some days, like Benedicta, and there are people that didn't even come at all, like Oguchi Gosri. So because of health issues. So these ones we still, through the video, learn and do the assignment and send back to to us. Then also why that is happening, maybe I would want to share my screen from my end. A moment. Yeah.
You see me?
Applications. Applications.
Am I showing?
Yeah. I have it in download already. I don't think so. It has to be from. Yeah, so where did I post this? I think it should be in Prime Groups. Okay.
So while we're doing that, Doc, please, you help us. Sending recording videos is very difficult to view those videos here. If you can put them on the YouTube so that we can just
we can do,
that
because it takes time. We need to edit before we put to YouTube. So, and the webmaster is quite deep. So we send the video so that we can continue our training. So after the training, we shall take time to edit and upload. Yeah, it's like it's better to do the uploading. It's like it's better to do the uploading than watching from the YouTube.
That's
So
the link.
yeah. So just so I think I can now share my screen. So as a continuation, and because I want many things to happen at the same time. So we on monthly basis also run what we call a webinar. So it started last month, where we started with applications and the essentials of bioinformatics. And next month, March 17th, so we should look at the prospects. So all of these should help to define some persons in an area, and do this over and over. Okay, so we invited Doc to do this because this is his major area. And we have people in sequences, we have people in maybe programming, very efficient in use of Linux and command line and app and many other areas in this. So thank God, of course, is an alma mater from UNIZIK. So, but currently is working as a bioinformatician in European Molecular Biology Laboratory, United Kingdom. So yeah, he will guide in this and present, and we shall take questions. Usually we don't spend much time. It's 45 minutes meeting and it's free admission, but you will need to register for this so that, yeah, you can participate, and we can also send you automatically the link for the meeting. So I failed to tell us so that we can get ready in the future to participate. Then the more you do this, the more you narrow. That is just the point. The more you narrow, the more you also wouldn't know some things are adding up. Now, most of the things that Doctor has been teaching, this we started 10 years ago in Faculty of Pharmacy, but a lot of things have really changed and improved, as I'm reviewing the course myself, because it's a review, now. So I think it will benefit us to be part of this and register. We shall still circulate this. Then for our WhatsApp group, we shall leave it for some time, but we shall provide you with a link to join the NIU Genomic and Bioinformatics WhatsApp group, where you get every information, because we don't want to multiply many WhatsApp groups. But this WhatsApp was to coordinate this training. Then thereafter, the deadline of submission of assignment, then we can close it down then and give you a link to join the proper website for WhatsApp group for the consortium. So the consortium, of course, is formed at Nnamdi Azikiwe. So the present Vice Chancellor initiated this with Professor Nwog of Parasitology and Entomology in Nnamdi Azikiwe University. So, and since 2019, we have been going on, though our inauguration was in 2019 by the Vice Chancellor, represented by the current Deputy Vice Chancellor Administration when he was a DAP. So he did the inauguration, and since then we have been pushing. So consortium means that there are many people come together, the MICTU of university, a Molecular Research Foundation, and the ProSeq also, and some other that thank God is working out in South Africa, the H3 Bionet also, that gives us opportunity. We would have held the NGS analysis, but we don't have people to serve as TA. So it's moved to Covenant University and Fountain University of Osun State, also in Osun State. So, but I know after this year training, maybe we could have 3 or 4 TAs that could serve in the future to host this around for us to participate. So we are still preaching about this evolving discipline and giving the young people. So within the year, we're also thinking and considering, of course, stepping it down again to those in final years so that they can start early to learn this, and they will not be doing school run at the same time learning bioinformatics. So, because if you don't learn on time, then some other things will drag your leg, where maybe later in the future when you're already busy with children, busy with many things. Yeah, but that doesn't mean that those who have children will not skate through, but you have less distraction in the course of this. So I will stop this slide because I will still send this to our WhatsApp group, and then you can, and also the clickable link from where you could now join and all of that. So I think that is the future information that we need to. And we also, just to round up, thank Samuel, even though the docking is still docking, okay? It's not properly docked yet, but let's do round up activities. Then he could do this and run out to enjoy his birthday with the children. Yeah, so as it's very important, if he doesn't do that today, it will be an issue in the house. Yeah, so maybe Professor will have to thank, on behalf of the participants, to thank Doctor. Yesterday, I was so much impressed that I have to send him personal. You know, this have taken my 3 days. Once we finish this, I don't usually achieve any other thing in the lab. I have to just round up and go. Yeah, but he himself is also very busy and have given us this time, detailed teaching, and from my capacity, I've appreciated and will still do that. But on behalf of the participants for the training, maybe Professor will thank him for us, and hopefully we'll see him again in the molecular dynamics. But if you don't practice molecular docking, dynamics will be a too advanced course for you. So because the basis of that is now docking. So once we go into docking, design a project, which will be the assignment you will do, and you scale through the assignment, then you are positioned for the dynamics. And once you're able to get a dynamics, this docking alone is enough to make you professor. A lot of paper that you can write with this, and we change project topic for our undergraduate students. They can do this. Yeah, so our students in Faculty of Pharmacy, they do docking perfectly. They can do this, but it's when we teachers learn that they can do. Okay, so things will change in the project we give students, and they'll begin to do what matters. Somebody should work on COVID-19 and check the local plants we have in Nigeria if anything can happen. And thank God is a research. If the result is positive, fine. If it's negative, fine. The issue is that you have researched and you have told the world what is the outcome of your own. It must not be positive. That's why in Germany here we have the Journal of Negative Results. So where you could bring in negative. So it's not any problem because it must not always be positive to reduce manipulation of data so that it will fit in and all of that. So yeah, so it must not always be positive. And also, Prof, please, could you thank our doctor for doing this? Yeah.
Let me, first of all, say thank you to you, our coordinator. You have been so wonderful since my interaction with you on this program. As I told you, my own is adult learning, and I will keep on pushing. Having said that, I also want to appreciate my colleagues in this workshop or training. They've done well. Some of them are already my consultants, whom I will go to help me fill in the gaps. Having said that, I want to, in a special way, thank our dear teacher. Let me use that word. He was patient enough to guide us through this molecular docking. He was always going back to address all the questions, never angry, never showing any sign that he's not comfortable. So I appreciate that, and I equally learned from that. It's not about learning molecular, you also learn how people treat those that are also mentoring or teaching. So Dr. Sam, thank you so much for this wonderful opportunity. Thank God also he has given you a new year today.
Amen.
I join our colleagues. I join our colleagues to say many, many happy returns to you. I know your family will be waiting for you, but tell them you also have a new family here today. So to cut the long story short, thank you, and thank you. And may God continue to bless you and give you strength and more wisdom and knowledge for his own glory and for the betterment of the world through Christ our Lord.
Amen. Amen. So thank you, Prof. Happy that you have been with us since last year. You know, yeah, it's an encouragement, and it's an encouragement, and I think we'll continue to have you on board as we do this. Yeah, maybe I will. Doc, is the running done?
Yeah, it's done.
It's done. Okay, let's take the final innocent Mary on behalf of the resource persons. Please, could you also make your remark? Then after that, Doc will show the last, and we shall say bye-bye. Then you can leave anytime you want, but we are going to have a comprehensive video which we are going to send back to us and with our assignment. So Innocent, you can unmute yourself and quickly say this closing ceremony, even though the docking is not here.
Okay, I want to start thanking Dr. Samuel for this wonderful 3 days of training, and also wish you happy birthday. And to the participants, you know, I have been doing docking, and every time I attend something like this, there's always something to learn. When you think you have known all in docking and you attend one program or the other, you will see that there's always something new to learn. So as Dr. Moses has said, when you go to your various departments or your various research areas and see how you can implement this, it starts from these little ones. And as you go on, you see that there are so many things you can do with this docking. So I thank you all.
Okay.
Dr. Moses, I just thank you so much for organizing this.
Thank God. Yeah. Okay. Thank you. And I think we should be back again to Doc finally, and he will interpret the results and make a conclusion. Sometimes you can stop your story in your paper at this point and make some kind of recommend further things that shall be done and get your data published. So yeah. So he will go on. So Sam, back to you again so that can move. Yeah.
Okay, thank you so much. I appreciate your comments, and I'm humbled by your greetings. Thank you so much. So I just want to finish up here. So once the run is done, you'll get an output like this view doc. That's what I was trying to open. I couldn't find my files. So it looks like this view doc. And on this view doc is a table. You have column X, core, RMSD, I.B and RMSD U.B. And you have this, let's call your attention to this score from minus 4.4. Let's close that off to 4.3, 4.2, and it goes all the way down like that. So that's the score. Now, what it does is that the one with the highest negative score is usually what we prefer, what we say most likely binding position, and the one with the what we call it now, highest binding affinity, as it were. Okay, so that's the way we interpret it, and yeah. So, but another thing that is quite important to do is to check the interaction, the number of interactions. We know that I spoke about weak bonds that is formed between the ligand and the protein. So we can check that by looking at this table. Let me try and bring this table to the center and make it a bit bigger, if I can. Okay, these are the biggest it can be. All right. So here we see this H bonds, that's the hydrogen bond. You can click on it and say, add the counts, that's the number of hydrogen bonds for this selected atom. Now I'm selecting this. Now you will notice I can go through each one of them. Let me just go through that. I can go through one. As I'm going through each one of them, it's changing on the visualizer. So that these are the different poses. So it's like you taking pictures and you're taking different positions. So this is what the computer has done. Try to put the ligand in the binding bucket in that pose. Okay, so Dr. Moses spoke about the ligand that was there before, which is the one I highlighted now. So I want to just hide that so that you can actually see what that one is doing. So I'm going to teach you a small trick because if you select A, you're going to select everything. Okay, so one small trick is, especially when you have a co-crystallized ligand in a model, if you want to select the either the protein alone or the ligand alone. So what you can do is to select, you go to selection mode and say, I want to replace this selection that I've made because the current selection is selecting everything. The current selection is selecting everything, both the co-crystallized ligand and the ribbon. But I can go there and say, I want this selection to replace I want to replace what I've selected now with what is currently selected with what I want to select now. Okay, I'll say that again. I want to replace what is currently selected with what I want to do now. So if I say replace and I go to select again, this time around I want to select only the co-crystallized ligand. I know its name is EH5. So it deselects the protein and select only the crystallized ligand. I can hide it. So go action, I'm not deleting, I just want to hide it. Action, atoms and bond and hide. So it's out of the way. All right. And I can also take away the grid box. I don't need the grid box anymore. Where's my doc thing? It's still there. No, I can't find that one. Anyway, I guess that's not in your way. All right. So I wanted to show you the different poses. Yeah. Different poses. You can see the different poses. All right. You can also, now what I want to do is to check the number of hydrogen bond that is formed between each of these poses and the, so I go to hydrogen bonds on this view log and I say add the number of counts, add counts to selected atoms. These are the selected atoms, such as that, add count. It brings out a dialog box. It's asking me, do you want me to find hydrogen bonds between intermodal, intramodal, or both? So intermodal means that between the protein model and the ligand model. Intramodal means that within that protein model itself. Okay, that's not really what I want. I want between the protein and the ligand, or you might want both. So depending on what you want, you can choose the color to display your ligand. Okay, you can choose a particular color if you want to. And you can choose the thickness of the line. You can label it. You can also, what else can you do here? Then this is most important, the distance. Okay, you can set a time, a distance. In literature, I think it's usually said that there are different, depending on the distance of the hydrogen bond. The strength of the hydrogen bond depends on how far on the distance. Let me put it that way. The strength depends on the distance. So the farther away it is, the weaker it is, the stronger the hydrogen bond in terms of the two interacting elements, the stronger it is. Okay, so I want to check if there are hydrogen bonds within 4 angstroms of two elements or two residues. Okay, and the degree is set such that how far do you want the line between two elements to be? Okay, that's what we're trying to set by the degree. Okay, so usually most papers have a standard way of values for these two. Okay, cool. And if I said okay, it's going to run that. Or did I say intermodal or intramodal? No, this has done for everything. No, I don't want this label. This label is confusing. I can't count. Let me do that again. Sorry. I'm taking away the model. And I only want to do no intermodal. Why is it okay, for some reason it's showing everything. I don't want everything. Apologies, I'm going to delete this one. I'm going to bring it in anyone. Let me see. This model. Oh, I see. There was another model. Sure. This was confusing the issue. All right. All right, that was why. So if I sit down, oh, okay, cool. That's all right. It's already built the model. Okay, so just look through this as I go through. You see the change in the current one has 2 hydrogen bonds. If you put your cursor on it, it will give you the details about the hydrogen bond. Okay, okay, saying that it's between the name of my ligand is UNI. Okay, the hydrogen on UNI and the oxygen on valine in the position 80 in that particular residue, and the distance between them is 5.353 Armstrong. Okay, if you do the same for to here, is between the element here is hydrogen that is in my ligand called UNI, and there's an interaction between that hydrogen and the oxygen that is on the aspartic at position 295 on chain A, and the distance is 4.7. Okay, so that's for the top. And if you go through the rest, you will see this one has only 1. This has about 3. The hydrogen one is in blue. This has about 2. Yeah. Remember now you are considering 2 things. First, you are considering the score. The more negative the score, the better the affinity. But we spoke about yesterday that it's possible that something binds, but it's not interacting with the right residue. And the protein can still go ahead and do its job. So not only do you want to check the affinity, you also want to check which residue is the compound binding to, being in that particular position. So that's also very important. Okay, we actually can create a movie of this, and it goes through each one of them, and you can just observe the hydrogen being formed, bond being formed. Once again, you can just put your cursor on that and see if it's interacting with the appropriate amino acid that you desired, or that has been noted in literature that is important for that particular enzyme or protein. Okay, there was one thing that I wanted to say. Bless you. Okay, now that's escaped me now. What I wanted to say. Okay, maybe I'll remember if there's any question. Can't remember right now. Okay, so yeah, like Doc said, we're gonna give you an assignment that will help you to engage with this. I'll try and formulate something that is, I don't know. If you come together with somebody, you should be able to put something good together. That you, yeah. And if you work out on it, you might be publishable also, at least to some extent.
To some extent, actually.
If I have not added molecular dynamics, if need be, we can add it.
Yeah. So now, what is the interpretation? Sorry, maybe I didn't get you. Yeah. So the best score here is -4.4.
Yeah.
But it has no data on the RMSD.
Okay. So basically, what they are trying to, the RMSD determines is how will I put it now? How the current pose deviates from what from a consensus configuration that has been done by the computer. So it has tried so many poses. You know, it comes up with a consensus pose, and it says this number 1 pose seems to fit that consensus pose more than any other person. Then the rest of the poses, it tells you how their deviation from that consensus pose. I don't know if I'm making sense. You know, so that's what it tries to highlight for each one of them. Now, people try to, in my view, that does, because it's a dynamic process, remember, we can't say that this ligand is going to take this position forever. That cannot be true. At a time, and that's why you're doing molecular dynamics. At one point in time, the ligand will take this position. At another point, it will take this position. Okay, so molecular dynamics is basically, if I do this movie now, play this movie, this is what molecular dynamics is doing. But we put all the necessary solvents and the rest, and create the right charges and the rest in terms of environment. But what it's going to do is that it gives the ligand and the protein the energy to move. So if you have given energy to the system, the ligand can assume any position as it deems fit based on the energy that is available in the environment. And it goes on doing that over time. Then what we tend to do is to plot the change in that position from the initial position that we started from, which is the 0 here. And we'll plot the change in that position with each pose, as it were, when it's in that environment. And then summarily, we can now say, putting all this position together that it has assumed when we stimulated in this environment, what is the most likely, not most likely, the word should be most frequent position that we will find this ligand. Okay, so if you do your RMSD, you will actually see a plot, an histogram, tells you that it will be in this position this number of times than it will be in this other position. Okay, so yeah, sometimes we make it oversimplify things just for us to say, okay, fine, it has the highest binding affinity, it interacts with my residue, fine. But if you're now subject to dynamics, you see that it doesn't stay in that position forever. It keeps changing, and we need to see how often does it stay in that particular position that I actually want. And I think that is the whole essence of doing the dynamics. All right.
Yeah, but if I could say, we have
sir.
1, 2, 3, 4, 5, around 8 score that came out.
Yes, sir.
And this is one ligand versus one protein.
Yes, sir.
So a situation we have many ligands.
Yes.
So we take a mean of this particular one, then each of them will come as a mean value.
No, it will not do a mean. So what people do is that when they have multiple ligands, they retain very few poses.
Okay, so the problem here is that we have many poses, but the best pose is minus 4.4.
Yeah, they just retain
the best pose and use it across. Okay, I see. Now, again, the experience I have in docking, I approach from command line, but I discovered that we have done everything in Chimera. Could you respond to this?
In what sense?
In what sense that after you assess your protein, get your ligand, prepare them, then through command line, you can run the docking.
Yes.
Yeah, but it's okay for this so that our participant will not be confused, because command line is usually confusing.
But
this is it's like an because what we did initially in the Faculty of Pharmacy was through command line. Then it also makes it to run faster. You know, Linus is faster than.
Of course not.
Yeah,
because you don't have visualization and everything.
Yeah. It runs and gives you everything. Yeah, so far so good.
You can run AutoDock on its own, AutoDock Vina on the command line.
Yeah, on the command line.
Especially when you have multiple ligands to run. This one is more for rechecking. If you already have a result and you want to recheck, this is more docking. That is virtual screening.
Fine. So I don't know if there's any comment from any issue from any person. Yeah. It's because
Okay. Let me just ask. So, Dr. Sam, can you please, in a way of summary, tell us how far we have achieved from the initial objective concerning this drug and ligand?
Okay. So in terms of what we set out to do, we want to know if this can also bind to the, what's the word I'm looking for, to this same receptor and inhibit it. Remember, we have a co-crystallized ligand which I hid. Let me unhide it, if I can find it. Which particularly has an interaction already. And if I go back to that interaction, let me go back here. Is it here? Oh yeah. Oh, just crashed.
Maybe, we could just summarize the outcome.
Yeah, but it's fine. So basically, what I'm trying to say is that it's that ligand that was with it from what I see in the initial, add more hydrogen bond interaction. So what I should do really is that I will take that ligand that was there before, I will prepare it the same way I prepared this and redock it and compare that interaction of that ligand with this one. So you have two things that you need to compare. You have two things that you must compare. What was naturally there, designed, and what you are proposing as a better one. Then you now compare the affinity and also the interaction.
As a term we used to define it, is like a baseline. Because if your own interaction is not above what is there initially, then it doesn't make sense. That is, if it is not stronger. If the binding affinity is not better than the, I've forgotten the term we used to, maybe Innocent can remind me.
The co-crystallized ligand.
No, when we have series of binding affinity, there is a cutoff. So the cutoff is usually what is known or what is being used initially. So yeah, for example, this ligand that is already binding on this protein, what is the strength there? So if your own is not stronger than that, then
it will always have competitive.
Yeah. So there's nothing to push for further on that. Yeah. So, but I think he's asking also what the summary is. For me, from my own end, we have been able to learn databases where to get protein, where to get ligands. Ligands could be chemical compound, ligand could be natural compounds. We have learned how to download them. We have learned how to prepare them also. Then we have also learned how to get our protein, from the biological and the compound from the chemical. Yeah, I think these are the two things we have learned day before yesterday and yesterday. We now looked at how to visualize and look at it. From my own table, I will say that I also learned how to trace the amino acid of interest from the primary structure. Even when it gets to the quaternary structure, I can still use the visualization tool to get there, so that if I need to dock that side, I should know where to put my grid balls, and all of that. So that we learned yesterday. Then today is now how to now dock to get a value of the strength of the interaction between the ligand and also the protein and all of that. So, and this is all done in silico, and you could also recommend in vitro studies if the in silico is good, or further validation. Yeah, but if it's now drug repurposing, then of course the drug is already approved, is a market. So you can now buy the drug that is meant for hypertension, but you could see that it can handle medicine-resistant orals. Then you can now do in vitro to see what is happening, and even while considering the side effects of the drug and all of that. So I think this is the summary of the whole picture. For me, then when you start working, playing around, yeah, you can now think out of the box and move on from there. Yeah, so James, I don't know whether it made sense at all.
Thank you very much.
Yeah, so Amara said that I have a question. You just ask the question. Okay, maybe use your voice, don't type it. You can unmute yourself. Amara, ask you to unmute, yeah. Amara, ask your question, go ahead.
This one matched. Okay. You better say go video throughout.
Huh? Amara, ask your question. Okay, so I think so far so good. So I'm going to get ready the video, maybe send the Zoom while we work on the website. And you quiz like you lost contact at a point, because I asked this question and it was responded to. Yeah, so the different scores you saw is because of different poses. Yeah, but you have to select the best, and the best is minus 4.4. So the lesser, the better. Yeah. The lesser, the better. Yeah. So I think Grace, you have a question. Yes, go ahead. Grace, you can unmute.
Okay.
Yeah.
Yeah. Sir, you made mention of applying docking in studying resistance in microorganisms.
Yeah.
So for instance, we have methicillin resistant Staph aureus that is resistant to multiple drugs. And you are trying to know if you can create another binding site in the organism to look out for new drugs, right? Am I right? So how do you apply docking in that sense?
Yeah. The point I was trying to I think I was coming from the angle of drug repurposing, okay? Now methicillin is failing and many other drugs might fail to some Staphylococcus aureus. So the question is, how could we get other drugs that may handle Staphylococcus aureus that are resistant? So in that case, you can decide to use chloroquine. So, but before you to do that, you may expose chloroquine to docking. You dock chloroquine with Staphylococcus protein, okay? And check whether it has a good binding affinity. This is just a layman how to explain it to you. Check if it has a good binding affinity. Then if it's showing in silico good binding affinity, then you can also try this thing again in vitro, okay? So in vitro means now using chloroquine in the lab to check whether it's going to inhibit. Are you understanding? And then from that stage, you can now move over to in vivo, try in animals or possibly try in human beings and see how the chloroquine. So it's a line of process to be able to now use another drug because other drugs are already failing to. Yeah, so I think that is just the summary of it. Thank you. Yeah.
Thank you, sir.
But it's important for you to also know what is causing the resistance.
Yeah.
So establish the mechanism of resistance. If it's a mutation in a particular protein, either the PBB or some of the other proteins, then you need to do quite a number of things, like Doc has said, in terms of trying some other things. But what is also very possible with molecular docking, and I think everybody can benefit from this, is that even when you're finished the docking and you see, for example, I dock penicillin against a resistant strain, and I find out the interaction. I find that it's not interacting with a particular thing, or it has lost affinity. I can speak to a medicinal chemist and say, can you add another group to this compound that I have here, such that an extension of this compound to this residue that is positioned here, or from what I see in this pocket is more hydrophobic. Can you make this compound more hydrophobic so that the affinity will increase? So now you get that knowledge from the docking. So the chemist can look at it and say, oh, if you want more interaction in this direction, I can do an optimization. So that's what we call it to lead optimization. And you can do that all in the computer before you even start testing the compound.
Thank you, sir.
Okay, so thank you. I think we have few people left now. Not few anyway. 16 is not few, but we have to end it here and say bye bye to
protein interaction. Somebody asking that you can visualize it. So it's also possible to visualize protein.
Yeah, he says, sorry, my browser is preventing access to microphone. Then protein interaction. Okay, preventing protein interaction.
Is it visualizing?
It's not clear to me. That is a Marana. That's why I say use your mic and ask yes, ask your question so that it will be clear.
I didn't get that.
I can one visualize a protein. Okay, so it's fine. So please, you can on your video so that I can wave all of us bye-bye and thank you for participating. Then please, you will soon hear from us and get our assignment and the video. Then take time again. We shall give you enough time to do the assignment. Enough time, because it will really be an assignment. Yeah. So if you could on your video, those that are still here, then we can wish you well and say final bye-bye to you.
Bye-bye.
Did I mute my mic? I request that we on our video.
Bye.
Atanda. Okay. Joy, bye.
Bye.
Yeah, Alimat, bye.
Innocent. I should hear from you.
Halimat, I hope we did not interrupt your going to mosque because it's already 2 o'clock. Pray for us, eh? Yeah, Chivo. Yeah.
Bye.
Yeah. Joy. Bye. Okay, permission to end, or you can leave now. Then maybe I will talk with Doc and the Innocent Mary briefly. Amarachi, bye. Bye. Yeah. Inosa, bye. So, okay, Benedicta, I know you have not been here. Bye. Chifo, bye. Grace, bye. Where is Doc? Why is your video not talking, Amarachi? You don't have audio. I will leave you with your problem, which are too much. Why is your audio not talking?